Hello everyone, I am Advait Anilal and I am here to present on our research titled Enabling Crop Stress Optimization of Z maize using an extremophyte driven comparative analysis. So to start off with, we all know that food insecurity is a growing threat in our world today. To combat this threat, new varieties of crops that can both handle abiotic and biotic stresses and provide greater yields are necessary. The insertion of genes from extremophytes, which are plants tolerant to various stresses, using CRISPR Cas9 technology, is an underutilized and potentially highly effective tool for creating such new varieties of crops. Genes from Schrenkiella parvula, which is an extremophyte tolerant of a variety of stresses, could prove useful in creating such new varieties of crops. To this end, we have conducted a comparative analysis of the proteome of this extremophyte with the crop Z maize, commonly known as corn. So our objectives are to identify genes in S. parvula that could be used in creating new stress-tolerant varieties of Z maize, and to identify potential gene targets for modification using gene editing technologies in Z maize. So the computational workflow that we developed is shown here. So we first extracted the proteomes of S. parvula and Z maize from UniProt with annotations describing their ontology and pathways. And then we used a Venn diagram maker to compare the two proteomes based on both gene ontology and the pathways that they are involved in. We then, from both analyses, extracted analogous elements in both species and elements found exclusively in S. parvula. So, for our results, for the analysis based on gene ontology, we found 65 common terms and 15 terms unique to S. parvula, as shown in this Venn diagram here. Some of the notable terms and their associated gene entries are shown in this table. This table only shows four gene ontology terms and their corresponding entries that are similar to both, and three that are unique to S. parvula because of a lack of space. For our analysis based on the pathways, we initially found three elements that were unique to S. parvula, as shown here, and four that were common. However, this was caused by differences in the way the pathways were ID'd between the two datasets, and all seven unique pathways in the S. parvula dataset were actually also found in the Z maze dataset. Some notable pathways are the peptidyl diaphthamide synthesis pathway, the malonyl CoA biosynthesis pathway, and the oxidative phosphorylation pathway. Coming to our conclusions, our research helps identify potential genetic targets for modification in Z maize. The dehydrin protein is especially notable here, as it is known to be involved in the response to the cold, abscisic acid, and water deprivation. The annexin NS3 gene is also notable for its role in the response to cold, heat, salt stress, and water deprivation. These are both found only in S. parvula. The computational workflow that we have described here could also be used on different species than the ones discussed here, which could help in creating new stress tolerant varieties of crops. Our analysis at present is, of course, limited by the fact that data on Schrenkiella parvula is limited. For example, only 7 entries in UniProt had their pathways denoted. Further research on this emerging model extremophyte is necessary to further refine our understanding of the plant and identify new genes that could potentially be used in creating stress tolerant varieties of crops. Thank you.